Okay, this is the second video that I'll have you watch, and what we're going to do is I'll spend the rest of the time going through a review of your exam, and then after Thanksgiving is where we'll pick up with Chapter 18. So your exam will go through 17, the material that we just covered in the previous video, as well as class. And let's take some time now to review this exam material for you. Now, the first question that I'm asking here is I would like you to be fully familiar with these key experiments about DNA. So things like the experiment by Frederick Griffith, in which he discovered the smooth and rough types of bacteria, and the fact that there is some kind of a transforming principle. The experiments by Avery and his colleagues that showed that transforming principle was DNA, and therefore the genetic material was DNA. And then third, the experiments by Hershey and Chase. And this particular question deals with those three. There's a fourth set of experiments I really want you to be familiar with. That's a big hint, by the way. And that's the experiments by Meselson and Stahl that deal with nitrogen 14, nitrogen 15, that helped show that DNA replication is semi-conservative. So make sure you're fully familiar with those experiments and what they mean, because I haven't written the exam yet, but you're likely to see a question on that, something like that on the exam. So anyway, this is an example of the sorts of things you need to know about those experiments. So again, the four experiments you need to know about are those by Frederick Griffith, Oswald Avery and his colleagues, Avery, McLeod, and McCarty, and then the Hershey and Chase experiments, and then the Meselson and Stahl experiments. Make sure you're fully familiar with what your textbook talks about on those experiments and the basic conclusions that everyone found from them. All right, so let's, this one essentially asks you to fill in a table that looks at each of these aspects over here and which of the experiments use these. One is they concluded that DNA was the genetic material. Well, Griffith only found that there was a transforming principle that was inherited. He did not know what the substance was. He was trying to find it when a German bomb hit his laboratory, and he was tragically killed as a result. Therefore, he never concluded that DNA was the genetic material, so you put no there. Avery et al. Then picked up on Griffith's experiments and designed a set of experiments that showed quite conclusively that whatever it was that the substance that was transforming R cells into S cells had to be DNA, could not be protein, could not be RNA. And therefore, they concluded that DNA was the genetic material. Hershey and Chase did the same thing, or did experiments with viruses, not the same thing. They did a different method. They used radioactive phosphorus and radioactive sulfur and were able to determine that the genetic material of bacterial viruses was DNA. So yes, they concluded that DNA was the genetic material. Neither Griffith nor Avery et al. Used radioactive isotopes. And yet Hershey and Chase used radioactive phosphorus and sulfur. So you'd say no, no, and yes for that. Did they use live bacteria? Yes, all of them did. Griffith, of course, used live R and S cells, and then he killed some of the S cells as one of his experiments, but of course he used live bacteria. Avery et al. Also did. What they did is they took different substances and added them to live R cells to see which substances would transform those live R cells into live S cells. So absolutely they used live bacteria. Well, what about Hershey and Chase? They used viruses. But the only way you can cause viruses to replicate is to have them infect live bacterial cells. So the answer therefore is yes, they used live bacteria. And D, used S and R strains of Streptococcus pneumoniae. That's what Griffiths was studying. Avery et al. Followed up on that, so yes, they used S and R. But Hershey and Chase were actually using a different type of bacterium. They were looking at viruses, not at the S and R strains, so the answer to that would be no. Griffiths didn't use viruses, worked purely with bacteria. Same thing with Avery et al. Hershey and Chase, on the other hand, were studying the genetic material of viruses, so the answer there would be yes. Now, in the second question, Oswald Avery and his co-workers used protein digesting enzymes and DNA digesting enzymes in their experiments. In other words, one set of experiments, they took various purified substances, added them to live R cells to see if they would transform them into S cells. But another thing they did is they would add an extract that was capable, stuff that was taken out of dead S cells, and then they would treat it with different enzymes to destroy whatever the transforming principle was, so that if it was protein, they could use some enzymes called proteases that will destroy proteins, and therefore if protein was the transforming principle and they added proteases to this substance before adding it to our cells, it should then be incapable of transforming them into S cells if protein is the genetic material. On the other hand, they use deoxyribonucleases, enzymes that destroy DNA but not protein. Therefore, if DNA is the transforming principle, if you add that enzyme to it, it should eliminate the ability to transform the cells. So that was their logic in this. And that's essentially what is explained in Part A. So B says, what are the results of the experiments in which they used protein digesting enzymes? Well, what happened is they used the protein digesting enzymes, they treated the extract with those. It destroyed the protein, but it didn't destroy the DNA. And when they added that substance to live R cells, they were converted into S cells. Therefore, protein could not be the transforming principle. On the other hand, Part C, the experiment when they use DNA digesting enzymes, when they add DNA digesting enzymes, it destroys the DNA, but not the protein. And when they did that, the substance could no longer transform R cells into S cells. Therefore, DNA must be the transforming principle and therefore the genetic material. Okay, now here's a question that has you put together a lot of different things. This will be material from chapters 15, 16, and 17 that you would need to use to answer this question. I really like, maybe you don't, but I really like these sorts of questions where it has multiple parts to them from a single research project that allows you then to interpret the data, integrating concepts that you've learned in a number of different chapters, and that's what happens here. In this case, we're looking at an article by Pryor et al., who described a mutant allele in the AR gene. What AR stands for is androgen receptor. That is, this gene encodes a protein which is a testosterone receptor. That is, the protein encoded by this gene, then testosterone will bind to it. When it does, it also has a DNA binding domain. Remember, this is the example I used in the previous video of a protein that has at least two different domains in it, one of them being the domain that is the testosterone binding domain. The other domain then is the DNA binding domain, because this protein functions as a transcription factor. It then, once it is activated by the testosterone, it goes on to bind to DNA in the nucleus and activate the genes that are responsible for the development of the male phenotype in humans. Therefore, if this gene is somehow inactivated, even if a Y chromosome is present, the individual will not develop as a male. And thus, certain mutations in it can cause what's called complete androgen insensitivity syndrome, which is means that individuals have a fully female phenotype but are completely infertile, whereas individuals with an XX chromosome constitution who are heterozygous for the mutant allele are fully fertile females with no androgen insensitivity symptoms. And in this case, in one of the families, they observed two full siblings who had an XY chromosome constitution. They were neither fraternal nor identical twins, meaning they were born at subsequent times. They had XY chromosomes, but they had that mutant allele on their X chromosome, and therefore they had complete androgen insensitivity, and therefore had a female phenotype and were fully infertile. The researchers then studied the DNA, and here is just a very small portion of the DNA of that gene. That gene has more than 800 codons in it. And these are just 4 of them that are near the end of it. They're in the 700, I think this was 773, I think is where is this codon right here is number 773. And that's where this mutation happens. And so I give you this information here. Here is the sequence of the RNA molecule written 5' to 3'. If you don't label it 5' and 3', you should assume that is written in the 5' to 3' direction. If you look at your genetic code, UAC encodes tyrosine, CGC encodes arginine, AUG methionine, CAC histidine. That is the non-mutant original, what we might call normal type, has that sequence in it. And then here's the mutated sequence, UAC-UGC. You'll notice that differs from the C up there. Everything else is the same. That should tell you, that will give you the clue that you need to answer some of these questions. So now down here it says, circle all the terms that apply to this particular mutation. Now you'll notice too, that there's something here that differs from your book. I told you in the last lecture that your book had a faulty definition in there, or one that wasn't quite correct. And that is that it refers to a missense mutation as one that just changes one amino acid. A missense mutation is one that can change any amino acid, and in fact, actually this one is fully in accordance, these answers are fully in accordance with what you'll have in your book, that it changes one amino acid. But a nonsense mutation, let's say one that creates a new stop codon, also alters the amino acid sequence, and therefore it would also be considered a missense mutation. Your textbook says that missense mutations will still code for an amino acid. That's not quite correct, because a nonsense mutation is also a missense mutation. So in this case, the answer here is fully in accordance with the definition in your book, where in this case it's a missense mutation because it changes the amino acid sequence. It is also a substitution mutation because what this one does is it substitutes. In this case it would be a CG pair in the DNA, and therefore it would substitute that CG pair with a TA pair. So in the RNA you have a substitution of U in the place of C. What that does is it creates a UGC codon which encodes for cysteine instead of arginine. By the way, as you've noticed, I like to use actual examples here. This is a real case in research of a mutation being documented that causes complete androgen insensitivity syndrome in individuals who are homozygous for it. It's in a gene on the X chromosome. Again, this brings chapter 15 material into it because you have to understand how X chromosomes operate here. So now it asks you to write dominant and completely dominant, codominant, or recessive to accurately describe the mutant allele. Well, how would you know what the answer to that is? What you have to do is go back here to the information, and what it tells you is that individuals who are hemizygous for this mutant allele externally have a fully female phenotype. Therefore, an XY individual with a fully female phenotype, that would be associated with that mutant allele. Now that doesn't tell you whether it's dominant or recessive because these individuals are hemizygous. They only have one copy of it. Therefore, if it were dominant, it would be expressed. If it's recessive, it would be expressed. So you don't know fully from that information what it is. But now notice what it says here. Individuals with an XX chromosome constitution who are heterozygous for the mutant allele are fully fertile females with no androgen insensitivity syndrome. That tells you if an individual is heterozygous for it, it must be recessive. Now there's an interesting thing here. I considered asking this as one of the questions. I don't think I put it in there. But would it be possible for an individual to be homozygous for this mutation? Think about that for a minute. For an individual to be homozygous for that mutation, that individual would have to inherit it from both parents. Because it's on the X chromosome, then that individual would have to have an XX chromosome constitution. And if that were to happen, we would presume, because of the recessive nature of the allele, that individual would have female phenotype. But it turns out that homozygotes are entirely unknown, and there's a very good reason for that. And that is, to inherit it from the father has to be an XY male. But if the father carries that mutation on, if an individual carries that mutation on the X chromosome, that individual will have a female phenotype, and therefore could not be the father. And for that reason, we don't see homozygotes for this, because males who carry this mutation aren't males; they have a female phenotype, and therefore, the females who carry it are heterozygous for it. They are fully fertile. They can pass it on to their offspring. But any individual who inherits the X chromosome carrying the mutated allele and a Y chromosome then from the father will have a female phenotype and be infertile. Any individual who inherits then an X chromosome from the mother carrying that mutation, that mutant allele, an X chromosome from the father then would be a heterozygous, fully fertile female carrier. And any individual inheriting an X chromosome without the mutation, in other words, the non-mutant allele from the mother, a Y chromosome from the father would be a fully fertile male. And then the other possibility would be inheriting the X chromosome with the non-mutant allele on it from the mother, and then the X chromosome with the non-mutant allele from the father, and would be homozygous for the non-mutant allele and would be a fully fertile female. Those are the four possible options. Therefore, the probability of an individual being born with a male phenotype, in this case, is 1/4, and a 3/4 chance of being born with a female phenotype, of which 1/3 of those females then would be infertile because they have androgen insensitivity syndrome. You can do a Punnett square to figure that whole thing out. So let's move on now to the other questions here. So from that information, we know that it's a recessive allele. Which parent contributed the mutant allele to the siblings? Had to be the mother, because if it were the father, couldn't be the father, because a father, an individual, I can't say the father, an individual carrying that mutation in the hemizygous condition is female and therefore can't be a father. And therefore the only person who could contribute this allele in this case would be the mother. The fact that there were two individuals means that she was a heterozygous carrier of it rather than having it be a brand new mutation. And so now, part D, how many bar bodies? The two siblings that have androgen insensitivity syndrome have only one X chromosome. Therefore, since even though they have a female phenotype, they don't have two X chromosomes, therefore they would have 0 bar bodies. The mother, because she has two X chromosomes, would have 1 bar body. The father, because he has only 1 X chromosome, has 0 bar bodies. And that's how you answer that one. Okay, this next one is a linkage question. You can expect a similar linkage question on your exam. This takes an example from 1916, Morgan and Bridges, who were determining the map distances in the X chromosome. In this case, they were looking at two different genes, FU and V. Mutant alleles in FU cause fused, if I remember the fused, I think the fused here is fused, the stripes on the abdomen tend to be fused. And then vermilion means that they have vermilion, very bright red eyes as mutant characteristics. That's the phenotypes associated with it. In this case, they crossed wild type females with vermilion fused males. All of the F1 flies were wild type. These mutations, by the way, are on the X chromosome. They allowed the F1 flies to mate with one another and observe the following in the male F2 progeny. Now, it's very critical that we're looking at the male F2 progeny here. Because in this case, interestingly, we expect on the X chromosome, if you do a Punnett square with X chromosomes, the males are going to be hemizygous, not homozygous or heterozygous, only hemizygous for their X chromosome, which they inherit only from their heterozygous female parent in an F2 generation. And therefore, we would expect a 1:1:1:1 ratio, not a 9:3:3:1. You can work out a Punnett square to see that. But That's what we would expect. If they were assorting independently, it would be 1 to 1 to 1 to 1. Pretty obviously, it's not. 394, 326, those are fairly similar. Notice those phenotypes are the same as the original parents. So it says here, in one experiment, they crossed wild-type females, there's wild-type in the F2 progeny, with vermilion-fused males, vermilion-fused. Those were the original parents. And notice those are the highest classes of progeny. The lower classes of progeny, these are the ones that arose from crossovers between the FU and the V genes in those heterozygous F1 females. So here are all the F1 flies that are wild type. Those females are heterozygous, crossovers can occur. What about the males? Hemizygous for the X chromosome and a Y chromosome. No crossovers, because there's no corresponding X chromosome for crossovers to occur. So you're not going to detect any crossovers there. Plus, even if it were an autosome in fruit flies, there are no crossovers in males anyway. The mapping has to be done entirely within the female parent, heterozygous female parents. And so these progeny right here are the ones that arose from crossovers between those two genes. That now allows us to map the genetic distance between them. And the way you do that is you take every individual who represents a crossover, divide by the total number of individuals, and that gives you the frequency of recombination, or the frequency of detected crossovers between those two genes. So in this case, that's how we would end up calculating the map distances. Before you do the map distance calculation, I want you to answer a number of other questions. One is FU, the fused phenotype, or that allele, is that recessive or dominant when compared to the wild type? Also the V one, is it recessive or dominant? Well, there's nice information in here that tells you exactly what it is. All of the F1 flies were wild type. Therefore, if they weren't, then we would expect, if they were dominant, then we would expect F1 flies to have the mutant phenotype, because each of them inherits that allele from their male parent. That is, each of the females, but all the females would carry the dominant type, all of the males would not, would be wild type, because the original female was wild type in the F1 generation. But in this case, all of the F1 flies are wild type. Therefore, both of them must be recessive, so you put recessive in both of these. And it says the observation that all F1 flies are wild type indicates that both mutant alleles must be recessive. Please, when you do your exam, remember to write the explanations whenever it asks for one, because I can at least give you partial credit for those, and even if you get the answers right, I can't give you full credit unless you write the explanation for me. Okay, now what are the phenotypes of the recombinant progeny? Now what this means is that some were fused and some were vermilion. If there were a comma between those, that means fused and vermilion together, or you can even write the word fused and vermilion. I'll try to make this, if I have a question like this, make it a little more clear so that you can distinguish between the four types of progeny. What this is meaning is that this is recombinant, this one is recombinant, this one is not, this one is not. I'll point those out here. Vermilion is recombinant, fused is recombinant, vermilion fused is not, and wild type is not. Now there are two ways that you can tell that. In fact, I ask you to tell me what those ways are. One is you look for the phenotypes that are the same as the original parents. That would be wild type and vermilion fused, because if you go up here, it tells you that in one experiment they crossed wild type females with vermilion fused males. Those are the two original parents. Therefore, those must be the non-recombinant types that you'll see in the progeny. And sure enough, the phenotypes that are the same as those of the original parents are the non-recombinant ones. And then here's the other way you can tell, is look at the numbers. The non-recombinant ones should be the most frequent, which they are, 394 and 326 compared to 126 and 143. The recombinant types then, fused and vermilion, are going to be the least frequent. So two ways to tell which are the recombinant and non-recombinant types. That is, see which ones have the phenotypes of the original parents. Secondly, see which ones don't. The original parents will be the non-recombinant. The ones that are different from the original parents are the recombinant types. That is, the phenotypes have recombined with respect to the parents. They also are going to be the least frequent because crossovers are less frequent than non-crossover types. And therefore the recombinant types will be the lowest numbers, the non-recombinants the highest numbers. You can tell that without even knowing what the original parents were. In fact, you can extrapolate to what the original parents were when you look at the data here, because the least frequent types will be the recombinant or crossover types. The more frequent types will be the non-recombinant or parental types, non-crossover types. And so very easy to figure out what the recombinant types are, and they must be fused and vermilion. Okay, and in part C, the FU and V genes are linked on the X chromosome. Describe the ratio Morgan and Bridges should have observed in the male F2 progeny if the alleles of these two genes assorted independently. In this case, because it's on the X chromosome, all of those males inherit their X chromosome from their female parent. And if it's assorting independently, the four types that are possible, which would be wild type, fused vermilion, then just fused, and then just vermilion, those are the four types that come out, should be equal because all four X chromosome types should occur in equal frequency in that female parent, and then all of the males inherit their X chromosome from that parent. And therefore, it should be 1 to 1 to 1 to 1, not 9 to 3 to 3 to 1. That's an important point that's only true for the X chromosome. If it were autosomes, then we would expect in an F2 generation a 9 to 3 to 3 to 1 ratio for independent assortment, but because it's the F chromosome, we expect a 1 to 1 to 1 ratio. If you have any questions or confusion about that, diagram it all out in a Punnett square, and you'll see that among the males, it will, even in an F2, it'll need to be 1 to 1 to 1 to 1. That's what we would expect. If it were an autosome, then yes, 9 to 3 to 3 to 1, but in this case, 1 to 1 to 1 to 1. Remember too, if it's an autosome and we're doing a test cross, that is, one of the parents is fully homozygous for those genes, then it would also be 1 to 1 to 1 to 1, rather than 9 to 3 to 3 to 1. Then determine the map distance. What you do is take the two recombinant type progeny, divide by the total number of progeny. That gives you your recombination frequency, which in this case is 0.271992. Multiply that by 100 to give you the distance in centimorgans. And I tell you here to mathematically round it to 1 decimal place, so that would be 27.2 centimorgans is the way that you figure the map distance. Okay, question 5 now. This goes to material from chapter 15, sorry. And it shows you a drawing of two chromosomes, one from humans, one from chimpanzees, and it tells you that there is some sort of chromosome rearrangement that distinguishes them, and you need to identify what it is. In this case, if you look at the two, they are the same up to this point right here. Then they differ, and then from this point down, they're the same. So it's this little region right here, right from about there to there, that they differ. But notice if you were to take the chimpanzee version and flip it over, it would be identical to the human version. Or alternatively, you take the human version and flip it over and it would be identical to the chimpanzee. And in fact, so it's an inversion. You don't know which species has the original conformation and which had the actual inversion. That has since been identified. It turns out that for chromosome 4, the inversion happened in the chimpanzee lineage, whereas humans had the original conformation. If it had been chromosome 1 and I'm trying to remember, and I think it's chromosome 18 have inversions in humans that happened in the human lineage. But this is one where it happened in the chimpanzee lineage. From this information, there's no way for you to tell. Could be one or the other, but you can very easily from this tell that it's an inversion, and therefore that's the correct answer. What would it be if it were a translocation? Then you would expect to see a completely different sequence on one end or the other, compared to that one. And likewise, the reciprocal rearrangement on another pair of chromosomes. So you'd have to look at 4 chromosomes rather than 2 to detect a translocation. A fusion, then of course 2 chromosomes coming together. A fission, where they split apart. It asks you here duplication, deletion, inversion, translocation, fusion, or fission. In this case, very obviously an inversion, because you just take that little segment, flip it over, and it would be identical. So an inversion is the correct answer for this one. And then I want you to explain that to me. You know, about 1/4, what I've said is about 1/4 of the chromosome, meaning from here to here is probably about, if you were to divide that chromosome before, that constitutes about 1/4 of the chromosome. That's how much was flipped over to make the inversion. Okay, last question. Within the telomeres of human chromosomes is the sequence TTA-GGG. We talked about that in class a little earlier this morning. That's the sequence that is added on to the end of a DNA molecule. And so it asks you here, it says within the telomeres of human chromosomes is the sequence TTA-GGG, repeated many times in tandem. Describe the function of this repeated sequence as it applies to the replication of linear DNA molecules. Be sure to use the term telomerase. As we mentioned, we discussed this in class. So what happens here is double-stranded linear DNA molecules cannot fully replicate on their ends, the reason being that there's a space left where the primer was, and the new nucleotides cannot be added onto the 5' end of that DNA molecule, and therefore it leaves a gap. What telomerase does is it goes in and it extends the DNA on the 3' end, allowing a primer to then be put on so that the gap can be filled in. And it's a way of preventing the erosion of the DNA molecule with each round of replication. So make sure you're fully aware of make sure you know this sequence, TTA-GGG, what it means and what telomerase does to make telomeres. Okay, that takes us to the end of the practice exam. This is the information that I will give you on your exam. I will give you the genetic code so that you can consult it. You certainly don't have to memorize what it is, but you do need to know then what the amino acids are in it. Notice too, let's go back to one of these questions here. Notice too that in asking you these sorts of questions, this is a common way to write the amino acids encoded by certain codons. If you use a 3-letter designation of the amino acid, for instance, tyrosine here, then you can put 1 letter above each of the nucleotides of the codon. So UAC encodes tyrosine, CGC arginine, and by putting a capital letter there and then 2 lowercase letters, it allows you to see exactly what the amino acid sequence is as it corresponds to its codons in the RNA. That's a very common way to write those two so that you can recognize them. That's the sort of way that I'll give it to you on exams and that I will expect you to be able to read it. Okay, just again, quick announcements. If you want to get in touch with me, send me an email message. Be happy to respond to that whenever I can. And I will be back in town. I get back late Saturday night, and so I'll be on campus on Monday, the day before the exam. The exam will be held in class on Tuesday, and we'll take it from there. So I will see you on Tuesday for the exam. Good luck on your studying. Also, I'm going to prepare a study guide of what concepts you need to study. I will be posting that a little bit later today. So we'll see you on Tuesday, and thanks for watching.